Thank you very much. Provost, colleagues and visitors, good evening. It's my great pleasure to introduce our speaker this evening, Professor Athanasios Mantalaris, as he takes the stage to deliver his inaugural lecture as the first holder of the Don Panos Chair of Pharmaceutical Biology, appointed in 2022, in tandem as a principal investigator in the National Institute for Bioprocessing Research and Training. Sakis Mantalaris is a man on a mission. In 2018, with some very clever strategic thinking, the School of Pharmacy and Pharmaceutical Sciences recognised that the biopharmaceutical sector, a critical component of the Irish economy, is shifting innovation focus from protein-based medicines towards more advanced therapies, including cell and gene therapies. So together with its partner NIBRT and the good fortune of the Don Panos Endowment, it was decided to create a new joint role, whereby the appointed PI would conduct cell therapy manufacturing related research at NIBRT, would conduct complementary fundamental cell research and clinical translational research at TTMI and St. James's Hospital, and would provide strong leadership and a strategic bridge between the institutions. Later on, many more partners were added. So anyone vaguely familiar with the Irish biomedical research ecosystem would realise what a massive task this would be. As a previous head of school told me, job descriptions are not meant to be taken seriously. If they were, no one would ever apply. But the School of Pharmacy knew what they were doing and diligently searched for the best talent available. They identified Sakis Mantalaris as the one who could fulfil this joint role and in the process lead the burgeoning cell and gene therapy ecosystem in Dublin and Ireland more broadly. Not much to ask at all. So Sakis Mantalaris is the man for the mission. But before coming to Trinity, Professor Mantalaris was Professor of Bioengineering in the Biomedical Systems Engineering Lab at the Wallace H. Coulter Department of Biomedical Engineering in Georgia Tech. Sakis obtained his PhD in chemical engineering in 2000 from the University of Rochester, USA, an alma mater we share. This led to the position of Governor's Lecturer in the Chemical Engineering Department in Imperial College in London. And subsequently, he was appointed to Reader in 2007 and Professor in 2010, also in the Chemical Engineering Department at Imperial. So Sakis has brought to Trinity his expertise as a senior leader in the field of bioprocessing and cell manufacturing. He has published hundreds of papers, as well as having developed several patents and spin-out companies. He has an impressive track record of independent research funding, having secured some $40 million from over 40 projects from agencies in the UK, the EU and the US, as well as from industry and charities. He has graduated 45 PhDs, many of whom are themselves in academia, and he was elected Fellow of the American Institute for Medical and Biological Engineering, has been awarded the Donald Prize in 2015 by the UK Institute of Chemical Engineering for his work on bioprocessing, and received an ERC Advanced Investigator Award in 2013, and many others. So evidently, Sakis has the academic credentials, the ambition and the vision for this demanding role. But as I mentioned, this is only half the battle. And I would like to quote from his colleagues. In his short time, he has shown extraordinary ability to bring people together from across the sector that wouldn't or couldn't work together in this area. The Irish Blood Transfusion Service and St. James's, the School of Medicine and the School of Pharmacy, NUIG and NIBRT, UCC and Maynooth, with a slew of industrial partners and government agencies to create Impact, the application which has been shortlisted for an SFI centre, and is the basis for a consortium that, considering the need, will have to be funded in some way eventually. Colleagues have recognised a personal authority coming from his academic standing, his personality, open, funny and engaging, his genuine passion for teaching and research, and obvious sense of fairness. He has shown that he's motivated by the common good. But not only that, but in an incredible stroke of good fortune and good sense, the School of Pharmacy and the School of Medicine were able to recruit Nikki Panaskaltis, Sakis' wife, collaborator, and I have a feeling much of his inspiration as well, the dream team. And Nicky, we look forward to your inaugural next year, so you will have the right of reply to any comments or remarks that he makes this evening. Now, Sakis is very well aware of the need to support students and younger researchers. He's been a kind and patient mentor to many, some 30% of whom have stayed in academia. He advocates constantly for support to academic colleagues and consistently attends every last school event, social and academic. He apparently loves a party and sees colleagues and friends and family to enjoy at work and socially. He definitely has a taste for the finer things in life. He's very proud of his Maserati that he drives too fast all the time, much to Nicky's embarrassment. Sakis is notoriously modest and uncomfortable with power and has a strong sense of fairness and social justice that drives his approach to his work and relationships. He is, however, snobbish about research quality and integrity. So to quote a colleague again, Sakis is impossible not to like. Well, maybe there's one thing. Sakis loves football and apparently was offered a scholarship to the US as a fullback. So I'll save you the trouble of reminding us about the other Greek team and the two recent matches between Greece and Ireland. So Professor Sakis Mantalaris, I invite you to give your inaugural lecture, Lessons from Cultivating Organisms from Bacteria to Humans.
Thank you. I'd like first to start by thanking Lina, because for 14 years I have avoided giving an inaugural lecture, and then thank the Provost, the Dean of Health Sciences, and everybody that came to attend this lecture. It's your fault. It was voluntary, so don't ask for any refund. There are 15 lessons that I thought were important to me. I didn't learn all of them, but I'll take you through a journey. And that journey begins in Athens. I was born and raised there many moons ago. And inadvertently, anybody who grows in Athens is influenced by the ancient Beatles or the Pops, you know. There is Plato, Aristotle, Pericles, and Socrates. And we learn a lot of lessons by them or about them and their work. The first lesson will be the last one, and I think it's an amazing lesson for everybody to learn. I attended Athens College. I was very privileged to go there, and I had a wonderful time while I was there. And our teachers were inspiring, fantastic people that had a mission in life. And I think that my passion about teaching started there. This is my graduating photo. I ran it through an AI vector index for image similarity. And clearly you can actually see that George and I are like identical twins. More importantly, I learned a lot of lessons, and the ones that I remember, and they're very vivid in my mind. The first one was, how will you take the tram if you don't know imaginary numbers? I can explain after the talk why that's the case. The next one was, I was taken back by the fact that in one of my maths exams I was given negative grades because my answers were stupid. I actually put a quote from Pascal, the French mathematician. It was actually at the back of every of our notebooks. Our teachers translated in, the more you talk, the more stupid things you're going to say. And the last one was, that I remember, was, it doesn't matter what you do in life as long as you try and be the best. I did learn imaginary numbers. I never got negative grades in school again, including university. I still talk too much. And I graduated number 7. had I graduated number 2, I could have been the current Prime Minister of Greece. So giving myself a solid D, I left Greece at the age of 18, and I started my North American adventure. So with no plan, I ended up in Canada at a specific university just because my cousin went there. But things happen for a reason. So I met there a very influential, probably the most influential academic mentor I had, the late Professor Argyros Margaritis. And Jerry, as he liked to be called, he actually said, don't waste your time with medicine. Come to engineering. And inadvertently, the lesson that I learned and I tried to apply, and of course to annoy Nikki, is that medicine of the future will be designed by engineers. I will not say the last part because that's going to annoy many people. So just to pay homage to Professor Margaritis, as we used to say at Georgia Tech, he was a hell of an engineer. I needed to go to the US. So Professor Margaritis was a UC Berkeley graduate. He wanted me to go there, but for other reasons, Nikki and I ended at the University of Rochester. And David Wu was my PhD advisor. I'll say one thing about David. He actually served his military service in Taiwan in the Department of Psychological Warfare. And I have many stories to tell you about David. But every time he would actually see me working on Christmas Day, he kept reminding me that AT&T Labs filed their semiconductor patent. These were the transistor on Christmas Day. He was lying to me. I checked that. So it was 28th of December. Nonetheless, the lesson that he would keep reminding me is that science is not a 9-to-5 endeavor. And that's a very important lesson, at least for me. But the most transformative experience was meeting Nikki. We met when we were in second year university, again by accident, and it was transformative because of two lessons. First one is always work with people that are smarter than you are. They will make you look smart. And I've applied that throughout my career. The next lesson is actually even more important because from then onwards, we actually started working together. So very early on, before interdisciplinarity became a term, it was cross-disciplinarity at the time. And interdisciplinarity has a specific meaning. We started working together. I'm not going to tell you an embarrassing story of the first paper we wrote together. Some of you know it. But nonetheless, it was a transformative experience, and it continues to be a transformative experience. So, 2000, we moved to Imperial, and for the first 4, maybe 5 years, we had the time of our lives. We were the 4 musketeers. I was Milady. And then there was Alex, Ferry, Manos, and Nikki. What an incredible environment. And again, I try to recreate it with Nikki every time with our own research group because it was really visionary. So the title is I Could Have Had a Career. So this is a 2023 paper. You all know about bioprinting of 3D organs. In 2004, we submitted a proposal printing 3D biomimetic constructs, spatial positioning of scaffolds, cells, and bioactive signals. The reviewer from the University of Oxford said it was the best proposal he had seen in 10 years, didn't get funded. Lonza launched a few years back their cocoon for cell therapy. In 2004, we had actually put in a proposal, modular bioprocessing technology for stem cell bioprocessing and tissue engineering, BioPro Lego. And we advocated a closed system for distributed manufacturing. A company that I won't mention, starts with an L, ends with an Onza, killed the proposal. In 2024, there was a paper, large-scale smart bioreactor with fully integrated wireless multivariate sensors and electronics for use in long-term in situ monitoring of stem cell culture. Again, in 2004, we had actually put in a proposal, intelligent wireless automated stem cell bioprocessing, talking about sensors, low-power electronics. It didn't get funded. 2004 was a significant year. It was the year that Greece had the Olympics, won the Euro, and won Eurovision. We got no funding. But we wanted to actually innovate in teaching as well. So I think it was 2005. You can't do everything. You can't have a completely bad year. So we continued. We put in a proposal to EPSRC and BBSRC for an alternative PhD structure that involved joint supervision, team, interdisciplinary PhD research team, and individual evaluation. And the response that we got back is stop messing about and do something serious. So I had fun. I would do it all over again. But I was naive. I thought that success was just straightforward. Not that I consider myself successful, but definitely success is not straightforward. So the lesson that I learned, I was not prepared to fail. This is now in hindsight and trying to be entertaining, but you will fail. At the very least, what you need to do is persist. So I could have had a career. Maybe I still have one. But I'll tell you, before I go to specific examples, what is it that I try to do? And I know that most of you, or a lot of you, will answer nothing, that I do nothing. That's not completely inaccurate, but I'll try and explain to you what my research philosophy has always been about. And it's reverse engineering. So if we look at the best bioreactor, and again, it is the womb, we want to reverse engineer what nature does at the very best. And I'll give you a couple of examples. I am obsessed with flying. So this is the American eagle. I did look about Irish birds. I found some, but there was not a lot of information, so I stuck with the American eagle. The information is from Wikipedia, so I don't know how accurate it is. But you will see, it has a particular length, wingspan, weight. The engines are 2 wings. Quite fast if you consider that it's a bird. Range up to 300 kilometers, so quite better than most electric cars. Quite a high altitude, fuel, dead flesh, protein, fat. And then when you look at the Airbus 380, it also flies. The principles are completely different. There are some, similarities in terms of length and widespan. Engines, 4 Rolls-Royce engines. Range, it's much, much longer. All I'm trying to say is that both birds and planes fly. Nature does it differently than engineering. We need to be honest with ourselves. We cannot say that, we want to mimic nature and biology and ignore the principles, and I'll explain that a little bit later. We are going to use engineering, so we need to be honest and do our reverse engineering as best as we can. I'll continue with flying. I'm not a violent person, nor am I militaristic. This is the Eurofighter Typhoon. But we are talking about cell therapy, so we do want to create a killer cell. So that's a killing machine. And just to give you a little bit more information about the Typhoon, its DNA, it's two twin Rolls-Royce XG40 engines. It can actually achieve over 2.5 Mach speed. DNA will tell you what can happen. This is again the engine, a little bit blown up, and you will see the cables. It's fly-by-wire. Think of the cables as proteins. You miss certain proteins, things shall not happen. You have DNA, you have proteins. If you don't have any fuel, nothing will happen. So this is the external tank of the, you can actually see it here, of the Eurofighter. It's actually energy that will tell you what will happen. So depending on the type of fuel, how much fuel you will have, actually that's what's going to determine the speed, the altitude, range, and everything else. So it doesn't matter that you have two twin Rolls-Royce engines. Unless you have the right fuel, you're not going to go anywhere. And for those of you who travel very frequently, you will know that sometimes you'll circle around while the pilots will actually empty the fuel so you can actually land. So metabolism is what I do. I'm a bioprocess engineer. That's the only thing I can control in a bioreactor, what I give into the cells, when I give it, and how I provide it. So if you look at a cell and you look at metabolism that controls gene regulation, cell proliferation, apoptosis, and everything else, and you consider the fact that cells do not grow by themselves, they like company, so you have cell-to-cell interaction. At the end of the day, what is happening is that there are certain inputs, modulators, cytokines, nutrients, metabolites, adhesion signals, mechanoelectrical signals. These are the things that you need to reverse engineer so that you can have a better cell culture. And by doing the right things, you can control proliferation, apoptosis, differentiation, and every other cell property, cell feature that you want in your culture. So not to sound as an estate agent, but it is the right signal at the right concentration, at the right location, at the right time. And in a nutshell, that's what reverse engineering is all about and what I have been trying to do. So I want to move now to the lessons, and I'll start with humans because I owe an immense debt of gratitude to the people that have gone through our labs. And this is not all of them. Unfortunately, they wouldn't fit in one slide, and I couldn't actually find all of these names. But as Brian said, Nikki and I have graduated successfully together, 45 PhD students, one that graduated after we left Imperial, and we've left one soul back in Atlanta that hopefully will graduate next year. 35 postdocs, over 50 MSc students, probably over 100 undergraduates. And it is because of the fact that they were really innovative, creative, full of energy, that this I consider to be the secret to eternal life. Associate yourself with young people, allow them to flourish, and you will always remain young. And I'm grateful to them. I will refer to Ioskani, who is my first and I think only academic grandchild at UCD. a little bit later when we talk about some of the work. So I'll start about bacteria, and I know that why would I have done work on bacteria? I'll explain to you why. I never had any interest. But this work is by Michalis Koutinas, who is an associate professor at Cyprus University of Technology, and Argyro Tsipa, that is an assistant professor at the University of Cyprus. And the reason why I started working with bacteria is funding. We moved to another phase of my career. We would write many grants. Still, the work that I wanted to do would not get funded, but work that I didn't want to do got funded. This is life for you. But in a nutshell, when you look at any microorganism, you have a substrate, and then that substrate is metabolized and the microorganism grows. And at that time, it was 2008, I was supposed to be on a sabbatical at Georgia Tech. My mother had lung cancer, so stayed back in London, visited Georgia Tech a little bit, but I did something that hopefully the provost will allow me to do again. I hid in the library. And it was good. I started reading. I started writing papers. So our collaborators wanted us to work on neural networks. I knew nothing about neural networks. I still know nothing about neural networks. I do know about imaginary numbers. So I said to Michalis, let's do something different. So the idea that we came up with was to rewrite Monod equation. So Monod, who won the Nobel Prize, was the first to capture kinetics, and basically the famous empirical equation that works says that the specific growth rate is linked to the maximum specific growth rate over a constant and the substrate concentration. Empirically it works fine, but you still have the process as a black box. So what we wanted to do, and the idea that I had, was let's look at gene expression and link it to substrate degradation and biomass growth and rewrite Monod kinetics. So we work with Pseudomonas putida, an amazing microorganism, very useful in wastewater, can actually metabolize toxic substances like xylene and toluene. And we identified genes, just to make it simple, that are linked to biomass and substrate consumption. I was thinking whether or not I should go through the math, but I won't. At any rate, what we actually did was we wrote equations for the genetic circuit. So we wrote equations for the genes and gene expression. And what happened was we were the first to actually describe that mathematically, but you can do real-time PCR, so you can actually look experimentally gene expression. And you may think that, look at the standard deviation and how accurate that is, but we were actually capturing the maxima, and we were actually capturing the trends. Any more granularity was not really required. So we knew that our equations of gene expression were correct, and the next challenge was to link microscale events, so gene expression, to growth and biomass growth and substrate utilization. So a little bit more math, and this is the Monod equation, and this is substrate inhibition, this is substrate degradation, and basically all you need to know is that your mother is also your niece, and your father is also your uncle. It's a very incestuous relationship. So what we ended up doing, we rewrote completely the monod equation. This is the specific growth linked to gene expression. Same thing with substrate utilization. And again, different genes, whereas in the monod equations, substrate appears in everything. I'm still waiting for Stockholm to call. Hasn't happened yet. Well, I only find it fair. He won the Nobel Prize for monod equation. We rewrote the equation. Anyways, one lives hoping. But we got a kick when we compare ourselves to the monod equation. So this is the simulations of the monod equation. These are the actual data, and the green line and blue line are our simulations. So we are using that approach to our stem cell cultures. But the lesson is that seize opportunities and do things your own way. So I had no interest in bacteria, but I didn't do what they wanted me to do. I wanted to do something that provided me with some satisfaction at least. I'll switch from bacteria to mammalian cells and the work of some key, because they're more involved, amazing people. I'll start with Cleo, the first PhD student that we started working on the modeling of these cells, and Cleo's PhD student, Ioskani, my first and only academic grandchild. He's doing amazing things, unfortunately at UCD. Don't take it personally, but nonetheless, he's doing amazing things. David Garcia, he's now director of the scientific directorate at Novartis. Alex Kiparissidis, who left UCL and an associate professor position to become an assistant professor at the Aristotle University of Thessaloniki. Greeks don't make sense to me, but anyways. Anna Quiroga, who is an amazing person. She's at AstraZeneca. And Antonio Grillo, who thankfully left Lonza and now is at WHO at the mRNA vaccine group there. So 2001, we were at Imperial, and Paul Nurse was awarded the Nobel Prize. Paul Nurse was awarded the Nobel Prize for work on the cell cycle. And why it was amazing was because he gave his acceptance speech before he gave it in Stockholm at Imperial. And it was inspiring. So we have a thing with Nikki where we like to exchange research insults at each other, and we challenge each other. So, you know, he started then challenging for other projects, why don't we model the cell cycle? So we looked at the literature, and what we found was that the modeling basically described cell size and cell age. And these are not qualities that are really meaningful and definitely don't provide you with any information. But we know that cyclin concentration oscillates. So with David Garcia, who still owes me his party in Colombia, we were going to go there for his bachelor party. Never happened. Anyways, at some point we'll do that. So we started saying, no, we're going to describe cell cycle by capturing cyclin concentration. And these are some partial differential equations. They're easy to write. Don't trust me, but they're easy to write because they're mass balances effectively. So, you know, entrance from previous phase, cell start at the phase, you know, change in time, change with cyclin, exit from the current phase. The difficult part is how you solve the partial differential equation. And that was above my pay grade. So we collaborated with some amazing chemical engineers / mathematicians in Greece. and basically they developed the discretization approach, whereby we don't lose information, but it's very computationally tractable. So again, mass balance is nothing really exciting, but the most important thing was that clearly with this approach we can get experimentally determined cell cycle parameters. Again, as George Box said, all models are wrong, some are useful. So we try to make our models useful by linking them with actual data and experiments that we can perform to validate. So these are the parameters that we could actually extract and validate, and we did the experiments. These are data that show cyclin distribution. This is cyclin B, this is cyclin E. So as long as you know which cyclins are important for your cell line, you can actually do the experiments, observe important parameters such as thresholds and minima, and you can then have a predictive model of the cell cycle. And we've used that in many, many different approaches. But what I'm going to show you is really the approach that we use to make cultures of cells that produce biologics better. So we developed a platform. I'm not going to go into details, but the thing to keep is that this platform really aims at precision bioprocessing, and that it is always experimentally validated. Actually, our approach is to reduce the number of experiments that we need to conduct and to have a predictive model. So what does that model tell us? It tells us a very important number. And this is cell type, cell line, clone specific, but it's the minimum number of energy that a cell needs so that it remains viable. If you drop below that minimum energy value, then the cell enters apoptosis. And I know we have Niall and Ioskani. They will clearly identify that this is exponential growth phase. The cells grow like crazy. That doesn't mean that they're not stressed. So if you then know what the energy is required, you can run an optimization algorithm. You put that as a constraint. And at the end of the day, what we've done is create our own culture media. Sorry, I'm a guy, so for me it's red, whatever you want to call it. This is the commercially available medium. The blue one is ours. You can actually see that we operate 10 times lower concentration of amino acids. So the commercially available medium is expensive. The more food you provide to somebody doesn't mean that it's good for that person. And in a nutshell, the next lesson was to tell you that on September the 11th, 2001, I was in the same train with a very famous colleague and senior colleague of mine. I was still a lecturer. We didn't know that the Twin Towers got bombed. He was asking me what I'm going to do with my life. I told him that I plan to model mammalian cell cultures, and he told me, and I quote, "You should do something better with your time." He was the PhD Viva examiner in one of my PhD students, because, you know, believe in yourself, but make sure that you prove yourself. So I will always remember that lesson. I'll switch again to multipotent stem cells, and this is work that we did with Nikki and some really talented people. Hugo Macedo, who is the CEO of his own spin-out company. Teresa, who is at the Karolinska. Mark Allenby, who 2 years ago became an assistant professor, or senior lecturer actually, sorry. I keep giving them promotions. But anyways, at the University of Queensland, and Susana Brito dos Santos, who is a research leader at now Saitiva. And my PhD was on three-dimensional culture systems of the bone marrow. And the bone marrow, and that's why Nikki and I work together, she being a hematologist, is an amazing organ. It's actually the best well-protected organ that we have in the human body because blood is created there. So when you actually look at the bone marrow, you will actually see that it has a particular three-dimensional structure. It looks like a sponge, and it has certain architectural and functional attributes that one needs to recreate. So we developed this three-dimensional hollow fiber bioreactor. We try to, again, engineer certain features so that the spongy material has the same porosity, pore size as the bone marrow. The hollow fibers will actually be able to allow cells and nutrients to go through. So we started culturing cells. And what is novel about what we did was, first of all, we could actually see that we could achieve cell densities that were close, and in some cases identical to actual bone marrow. I wouldn't say identical, but close. I'll stick with close. We could actually have red cells transverse the hollow fiber, exactly as what happens in the bone marrow. We could actually generate enucleated red cells. And we did some, again, by necessity, things so that we can really explain the results. We developed our own image analysis code so we can actually extract a lot of information and see what is happening in these cultures. And what we noticed was that these cultures were actually generating their own stroma cells. Stroma cells are the cells that support hematopoiesis. They provide the growth factors that the cells need to grow. And we verified that, indeed the cells will provide, maybe it's in another slide, they will actually provide the growth factors. So at the end of the day, we had a system that could generate red blood cells, and we could actually produce, for a very small volume, 1 billion enucleated red cells in the absence of exogenous growth factors. And I'll say that because you take the same cells as starting material, you put it in a flash culture, and the cells will die in about a week. So, with John's blessing, I can go back into the lab with Nikki and, do really the things that we like. But that brings me to my next lesson, second to last, which is identify nature's critical quality attributes and mimic them. So don't think that you are mimicking nature when you're just doing what you want with your cell cultures in a two-dimensional flask where you provide none of what nature does. And I'll finish off with my pluripotent stem cell work. And I was blessed that certain events in the US that had to do with the election of certain presidents meant that the Korean pipeline would not go to the US and came to the UK. So some amazing Korean students, Yusik, who is now a professor at Kyunhee University, and Jae Ming, who is an associate professor at Incheon National University. And I had a fascination during my PhD. My advisor and I, he was in Japan, I was back in the US. Because of the time difference, we could basically work 24 hours. So I would work, I would give him things. When I would go to sleep, he would actually read them, give me feedback. And we wrote a grant to NASA, and it got funded. So I was really taken by NASA's perfusion bioreactor. This is supposed to be a simulated microgravity bioreactor. It doesn't simulate microgravity. Microgravity is 10 to the minus 6. They can only pull 10 to the minus 2. But at any rate, I managed to get a little bit of money, and we got this perfusion bioreactor in the lab. And we started using it, and it didn't work. So there is a lot of work behind it. We actually did both simulations and experiments. We identified the reason why their perfusion bioreactor did not work. It was basically basic engineering principles. We created our own, as you can actually see here, and we created a system whereby we could encapsulate in little hydrogel spheres embryonic stem cells, undifferentiated, put them in the bioreactor, and it's load and forget. As in, and now that I think about it, they should have done more work because you don't need to feed the cells. At any rate, we could actually then differentiate them in whatever we wanted. And the next slide, again, it has mathematics. It's just simple mass balances, really. Nothing overly complicated. But basically, we developed a model. We actually linked the things that we learned from the Monod equation. And what we actually showed was that perfusion would remove the metabolic stress that cells experience in normal cell culture, even if you're feeding your cells every 2 or 3 days. So we noticed that accumulation of metabolites affects growth, and it primarily affected the quality of the cells. So that was extremely interesting. This graph shows exactly what I mean. You actually have here glucose. It's a nutrient. In a normal batch culture, it is quickly exhausted. More importantly, you have lactate, which is a toxic byproduct of metabolism. It's accumulated. Same thing with glutamine, nutrient, ammonia, a toxic byproduct. In a perfusion system, even if you don't optimize it, you don't see these extremes. And we were able, not mechanistically, but experimentally figure out why the cells were not of the quality that we wanted. And we showed that this toxic metabolite accumulation basically downregulated certain receptor genes that are critical for a particular signaling molecule to make the quality of the cells remain really high. So you can close your eyes. We actually did some animal experiments. We took 10 of our hydrogels we could produce in a 50 ml bioreactor, about 500 of them. They put them in a critical size defect. Critical size defect means that the animal, the patient, will never be able to regenerate on its own, on his own, her own, the defect. And basically, even though they only put 10, they could have actually packed far more. We could actually see the fact that we had de novo bone synthesis at the center of the critical size defect. Had they allowed the animals to go longer, we could have actually got complete healing of the critical size defect. I loved NASA. It was a dream of mine. So my last lesson is pursue your dreams and your ideas. there is a price to pay, but it's worth that price. So I told you at the very beginning, the Paps and Socrates was really a lesson that was driven to us, but I think it's an important lesson for everybody. True knowledge exists in knowing that you know nothing. And I do know nothing, but I think that's the only way to keep improving. So that brings us to our Irish years. A very unfortunately influential person, John the Greek Gilmer. I'm going to say some nice things about John, so please plug your ears, John. So back in 2021, January, John and I started talking. And he had a vision. He had a vision not just for the position. He had a vision for how to link NIBRD, how to link TTMI. He had a vision that we should try and do something national. And with John and Nikki, we started to develop that vision. So then I talked to, and I quote, John's better looking and wealthier cousin. And we started developing what you will see is now starting to materialize. Because we have been very lucky to put together a group of extremely talented people, and my last hope of trying to learn something. So we have, Liz, Archita, Michelle, Diogo, our experienced researchers, and we have Ocean, Eva, Corinne, Miriam, and soon to join us in March, Suman and Kostas, creating the lab that Nikki and I studied at Imperial, moved to Georgia Tech, and now at Trinity and NIBRD. We're lucky to get the SFI Research Professorship. And unfortunately, instead of spending time in the lab and with an amazing group of people, we are working with, again, a group of outstanding people at Trinity St. James Children's, and of course NIBRD, and throughout Ireland on the Irish Medicine's Personalized Advanced Cellular Therapeutics Center that we hope to get. So I just have a few slides to give my thanks. There are the funding agencies, and I know that I have missed a few of them, but we were lucky in our career to be relatively well-funding, apart from the first 4 years. There are some amazing colleagues that were influential, at least in what I did. I start with Steve Asprey. We started working on the modeling together, but he realized that there is more money in the dark side. So I'll leave it at that. Alex, what can I say about Alex? Apart from we would sleep at Imperial without anybody knowing, because you had to vacate the building at 11 p.m. We would sleep in a rented accommodation when it was snowing and we couldn't actually go home. Then we would make it to our house. Ferry, what can I say about Ferry? We were actually going and emptying Alex's office, telling him that he has been fired. Manos, our first grant together, we violated BBSRC's deadlines. We actually drove and hand-delivered the grant ourselves. And the late Dame Julia Pollack, we had a love-hate relationship. She loved to hate me, but she was a great scientist and amazing mentor. I told you about the Greek chemical engineers that solved the equations, Michalis Georgiadis, an amazing scientist. And from my Georgia Tech days, Stas Emelianov, because we are planning to take that bioreactor. I had actually put ultrasonic devices there to provide mechanical stimulation. We're looking at piezoelectric hydrogels, and the idea is to try and grow meat and hopefully send the bioreactor and Elon Musk to Mars together. So we're just putting a proposal soon. I do have a debt of internal gratitude. So in Greek, everything bad is masculine. Tax, war, division, death. And in Greek, everything good is feminine. Life, love, happiness. Sophia means wisdom. I apologize about that. So my internal gratitude goes to my late mother, Nikki, my partner, because they did give me life, they did give me love, they did give me happiness. They failed at wisdom. So I give them a solid B. And my last thanks is to you. I did put you through a lot, but, it was voluntary for you to come here. Thank you.